Hello, students, can you hear me? Okay. Take out your PDF, okay? Mineral nutrition or PDF already forward kora hoise, please take it out. We'll have a discussion on the lesson. We don't have to write anything because entire thing is written here. Okay, so I think we can start the chapter. Before going into the chapter, let's know what is the type of question that is asked from this chapter. In this chapter, we have to talk about the different types of minerals that are present inside the plant body. All the minerals that are present inside has been divided under two categories, that is the micronutrients and the macronutrients, depending on what? Depending on the amount of concentration it is present inside the plant, right? Jeti basic ase, that is what is called as the macronutrient. Jeti kom amount ot ase, tak ami ki boli kom micronutrient, right? So etu chapter ki pori macro or micronutrients or details like deficiency symptoms, right, where they are found, in which part of the plant body they are found, constituent key, etc., etc. So if you have gone through the PDF and the notes, you will see that we have given some information about certain mineral elements over here. Tar bitorte deficiency syndrome or upor basic question ahe, right? As far as the constituent or maybe where the mineral is exactly found in the plant body, ebilakor basic questions aha dekha nai. Questions are that maybe nitrogen deficiency hole plant edal or ki hobo, right? Or maybe these are the deficiencies. These deficiencies could be due to the lack of the these are the symptoms. So these symptoms could be due to the lack of the deficiency of which mineral element. So deficiency column to ami bhalke pohim tare. Next is tar bhitor to ki poribo lage, we'll tick them out and then important ami mark korim. Apart from that, ketaman terms janibo lagibo, jeneke as I said first, micro and macro. What is micro, what is macro? Next is another two terms, that is deficiency and toxicity, right? What is deficiency, what is toxicity? Deficiency, as the name suggests, we know jiman amount ase tatke jedi komot lage, no, whatever the amount is required, if it's lesser than that, then it's called as what? Deficiency. Whatever the amount is required, and if that is more than that amount, that will cause what? Toxicity. Jiman ase tatke kom, deficiency, jiman lage tatke besi, that is toxicity, right? So toxicity report, just few lines we have to remember, whatever there is there in the PDF, these are the only lines that you will study. আৰু একো বেছি নালাগে, ঠিক আছে? So কিছুমান elements toxic হয়, and generally when we talk about minerals into micro and macro, generally the micronutrients are more responsible for causing toxicity inside the plant body. But what is the reason behind? Because toxicity hobo lagile jiman amount ase tatke okoman besi hobo lagibo, titial tak ame ki boli kom toxic. Micronutrients bilakor eneo amount to ki, it's very less in the body, the requirement or the amount in its present inside the plant body is very less. So okoman due to certain okoman kiba ta beleg factor or cardio ki hoy, okoman ke increase jodi hoi jay, then it will become toxic. So toxicity is generally related with which group? Micro and macro in general, micro in more or in general, right? Next, adult plant, if there is a toxicity, then it will lead to some other symptoms also. So yate amar ketaman symptoms ase, hegita ami porim, eta specific disease ase, that is also we are going to read. Next, apart from this macro and micro, we'll have to talk about nitrogen fixation, right? Nitrogen cycle, nitrogen fixation poribo lagibo. Talking about nitrogen fixation, again some of the things that we have already studied, no? Symbiotic nitrogen fixation, asymbiotic nitrogen fixation. Nitrogen fixation could be physical, it could be biological also. So examples bilak lagibo. Next is when we talk about nitrogen fixation, in details what we are going to study is that leguminous plants, Rhizobium and leguminous plants with root nodule formation, etc., etc. So questions, duta ba tinta question from that part, right? Just 3 points that we have to study, not in details, right? Jetia nodules form kore, which cell is responsible for formation of the nodules? Nodules mane ki, nothing but a collection of cells, right? A group of cells is called as nodules. So kuntu group e divide, kuntu cell e divide kori and redivide kori pelai, hey nodule to form kori, so that is something that we have to know. How the rhizobium enters inside the root cells, that is another point that we have known. There should be certain types of attractions, right? You have the root in the soil, you have the microbes in the soil also. How will the microbe know which plant to affect? right? Soil to okol leguminous plant nathake no. We know whenever we talk about symbiotic nitrogen fixation, we talk about leguminous plants. When we talk about leguminous plants, we talk about the link between rhizobium and the root nodules and the leguminous plants, right? Now, where will you find the rhizobium? In the soil. Where will you find the leguminous plant? Obviously in the soil itself. That does not mean that particular area of soil had only leguminous plants. There might be so many different types of plants growing in the same soil or in the same area. How will the rhizobium know which plant to affect? Kuntu plantor bhitor rhizobium to humabo lagibo, hetu to janibo lagibo no. Rhizobium to kot jabo, root nodules form koribole leguminous plants or bhitor jabo. Baki plants or bhitor kyo na jay? There are certain types of chemical secretions, and because of these chemical secretions they get attracted and ultimately they enter. What they do there? Do infection cause kore, infection thread bully eta word ase. So these are the things that we have to talk about. Apart from this, the next part is, since we are talking about nitrogen fixation, plants will absorb nitrogen in which form from the soil? In the form of nitrate. Some of the part we have already done, no? Nitrification part to ami korisilo, biological classification. I think you remember something called as nitrosomonas group and the nitrobacter group, right? This is a part that we have done. So, tar bhitor ki questions ahibo, je ammonia to nitrate kune convert kore? Nitrite to nitrate kune convert kore, right? Ammonia to nitrate, nitrite to nitrate, the group of bacteria that is involved. That is one common question. Next is, once the nitrate form is absorbed by the plant body, that nitrate, what will happen to the nitrate inside the plant cell? It will get reduced again. Nitrate to ako ki hobo? Nitrite le bhangi jabo. Nitrite to ki hobo? Ako ammonia le bhangi jabo. Means what is the sequence I am talking about is, nitrogen fixation boli kole, we'll start it from atmospheric nitrogen. Atmospheric nitrogen will form the various usable forms like ammonia, nitrite and then nitrate. Kot form koribo, soil form koribo. Roots bilake ki lobo? Nitrate to humai lobo, right? But that nitrate cannot be used as such. Nitrate to ki koribo laibo, ako convert koribo laibo. So all the reactions that were occurring inside the plant, outside the plant body, ebar ki hobo? Bhitor hobo. Bhitor cell to ki ase etia nitrate ase, nitrate to ki hole convert hobo? Nitrite. Nitrite to ki hole convert hobo? Ammonia. What will happen to that ammonia? That ammonia, with the help of combination with different substances, will ultimately form ki amino acids, right? So atmospheric nitrogen is present in the form of amino acids in the plant body. Nitrogen to low pelai ki hole convert hole? Amino acid. Amino acid kune form korile? Ammonia form korile? Keneke form korile? By combining with different products that are present already in the cell, right? That is another point. Next point is talking about amino acids. Another point that we have to know. We know that amino acids bilake ki kore? Etar pra etale beleg form kori thake. 20 amino acids are there, and then they can combine with different things to form new amino acids, right? So as a result, the statement that we say is amino acids are not stable. So jihetu stable nohoy, amino acid bar thik ase, nitrogen to amino acid hoy gol. Amino acid is stable nohoy, hi ki koribo? Beleg products form koribo. So what is the stable form? We will be talking about amide, right? So amino acid bilak ki hobo? Amide ole convert hobo. Amide is the most stable form of nitrogen that is present inside the plant body. Tar bhitor question ahe, what is the first form amino acid inside the plant body? What is the first form amide inside the plant body? That's all. Right? And lastly, what we have is next is a little bit about absorption. Since we are talking about mineral nutrition boli kwa loge mineral to absorb koribo lagibo no plant dale. So wherever we talk about mineral absorption, either it could be active absorption or it could be passive absorption. When we talk about passive absorption, process ki ATP na lage, ki process? Diffusion, osmosis, etc. Ebilak processor karne jedi hoy and high to low concentration jedi hoy, tak ami ki boli kom? Passive. If it is just the opposite, lower concentration to higher concentration goise with the help of extra ATP, tak ami ki boli kom? Passive. So absorption keneke hobo? Either active or passive. Absorption hoar pisat keneke circulate koribo? Along with the water, that is through the xylem. And ultimately we will see the different parts where the micro and the macronutrients are present. That's all. Aru tatke eku nai. Aru eta bostu ki ase? Je talking about these different types of nutrients that are present inside the soil. Adult plant tot on kora nai. Bondo hoi gol. Janu video aha nai. এক সেকেন্ড হা এটাও নাই যোৱা নাই তুমি না এক সেকেন্ড হা শুনাটো পাই আছা ন PDF গিটা আছে ফ্ৰণ্টত okay okay next is another point that you have to know is something called as essential elements first we know inside the plant body ki thake minerals thake minerals ki korise divided into two categories micro and macro right now the point is how do you know that inside the plant body there are certain types of mineral nutrients plant or bhitor je minerals ase ami keneke gom palu কিবা এটা টেকনিক থাকিব লাগিব ন we cannot be just going into a plant and plant to দেখাৰ লগে লগে তো আমি গম নাপাও so that minerals so we talk about something called as ash analysis right what you do you dry the plant you burn the plant burn কৰাৰ পিছত কি থাকিব সাইখিনি থাকিব না থাকে ash right you study that ash and that ash analysis talks about the presence of different types of micronutrients and macronutrients inside the plant body out of all the macro and micro jiman bilak nutrients ase around 70 of them out of them we have a word that is called as essential nutrients asile no essential nutrients kiman asile according to the NCERT kisuman books of 16 ase kisuman book of 17 ase kintu NCERT ki ase 17 so we'll be sticking to that point right so plantor bhitorot how many essential elements 17 out of all the nutrients which element would be considered as essential প্ল্যান্টৰ ভিতৰত বহুত নিউট্ৰিয়েন্টস আছে কিন্তু তাৰ ভিতৰত অকল 17 টাকে আমি কি বুলি কৈছো এসেনশিয়াল so on which criteria will you choose which element is essential we have to study something called as criteria for essentiality right eta elementor ketaman conditions thakibo if they fulfill those conditions then that element or mineral would be considered as key essential so what are the conditions it's already given in your NCERT book plus it's given in the notes also right kenukwa dhoroner conditions he element to nohole plant dal mori jabo laibo right he element to deficiency kuneo belege solve koribo nware if a plant is suffering from suppose deficiency of x you have to apply x only y apply korile tar deficiency symptoms heri nohoy cure nohoy right another condition could be a deficiency of a particular element could affect the vegetative phase of the reproductive phase of a plant so enukwa dhoroner conditions ase those are called as essential criteria or criteria for essentiality of an element hekhini poribo lagibo kune disile that's all right so what we'll do hekhiniye hoy tar uporot whatever I have said what is the most important question that is asked is the deficiency symptoms of the different macro and micronutrients aru eta word mobile elements aru immobile elements basic details eku nahe you have to identify which one is a mobile element you have to identify which one is the immobile some of the mineral nutrients are mobile boli koise why is it called as mobile because it moves from one place to the other place right are immobile pur jote synthesis hoy tate thaki jay that is called as immobile so ketaman mobile ase ketaman immobile ase question to keneke ahe identify the mobile another question associated with that part is mobile elements bilakor deficiency hole kuntu part of the plant body affect koribo immobile jedi elements or kotha ko kuntu part of the plant body affect koribo younger part and the older part and as we go along we'll be marking it down just go to the first page if you are talking about mineral nutrition obviously minerals would be taken in by the soil hetu ami janu then we have talked about the ash analysis what is ash analysis dry koribo burn koribo ash khini pam ami study korim he ash tray ki koise the presence of various types of minerals inside the plant body right the question ahe how did people come to know the presence of different types of minerals inside the plant body the answer would be by doing of analysis of ash by ash analysis okay about 50 to 60 elements are present in the plant body as we have said tar bitor ki hoy je Arnon and Stout proposed the criteria for essentiality of elements as I said out of 50 to 60 or 70 elements only 17 elements are said to be essential essential keneke hobo kisman criteria thakibo hey criteria bila kune disile two scientists called as Arnon and Stout so Arnon and Stout e ketaman rules disile hey rules kita jodi follow kore tetale element to ami ki buli kom essential buli kom right so what are the rules actually it's better we'll study it from the NCERT book tate okon beshi bhalke dia ase yato ase the element must be necessary for normal growth and reproduction right the requirement of the element must be specific for the plant life The elements must be directly involved with the metabolism of the plant. The absence or deficiency of the element produces disorders. These are some of the conditions for essentiality. Egita jodi condition hoy tetiahole tak ami ki buli kom essential element buli kom, right? So what is the question that is asked? There might be two questions. First, who has given the criteria for essentiality? Name of the scientist important. Second is maybe kisuman options di dibo and you have to identify the essentiality factor. Hattar birot jikono eta correct hobo, right? Okay. Next, 17 essential elements. 17 essential elements divided into two types, macro elements and micro. You have to remember the macro and the micro. Suppose the carbon is an example of a macro element and a micro element, right? Think like boron is an example of macro and micro, right? So out of the 17, no, okol jodi macro gita if you by heart, then obviously apart from those macro, everything would be micro, right? So what do you remember? CHON, SPK, MgCa, right? Right? So ekhini bostu ki hoy? Macro, baki GA diok. Baki bilak ki hobo? Micro. So you remember this part. My way of remembering is, if suppose we are talking about this one, it's almost like a question mark, no? Hindit kon buli kole ki jai? Question mark hoy no hoy? As if you are asking who is SPK and MgCa. You take out your own tricks. My way of remembering is that I just remember it like this. As if I am asking a question, SPK kun hoy or MgCa kun hoy. Kon buli kole to kun hoy hoy jai, no? So you can take out your own tricks to remember. But the point is you have to know the macronutrients, right? Macronutrients jodi mukhosto kora, micronutrients jodi mukhosto nokorileo hoy jabo, right? It is suppose Cl jodi ko macro hobo ne micro hobo, obviously micro hobo karon yato nai. So etu ki hobo? Micro, right? So please by heart these. Okay. So concentration must be more, concentration must be less. Concentration to okman saba ketiaba question ahibo pare, not a very frequently asked question in tothapi. 1.0 to 0.1 in case of micro, 1 to 10 mg per liter in case of macro. Kintu what is the frequently asked question? The name of the macro and the name of the micro. Next, apart from these essential elements, there are certain elements that are put under specific headings. For example, certain elements are called as protoplasmic elements. Why they are called as protoplasmic elements? Because they are responsible for the formation of the protoplasm, right? So protoplasmic questions ahibo pare, identify the protoplasmic elements, identify the antagonistic elements, identify the balancing elements, right? So eneke jikita heading dia ase hekita poriba. Out of this protoplasmic elements or question ahise, then membrane permeability upor question ahise, some minerals that will help in deciding whether the membrane is permeable, impermeable, semi or selectively. Next, certain minerals that are called as activators of enzyme, they are responsible for what? For the functioning of the enzymes. Next, beneficial nutrients, mineral elements other than the essential elements which satisfy specific additional nutrient requirement for some plant. Tar mane kisuman elements of ami ki boli koiso beneficial boli koiso. Beneficial mane they will help, they will give extra help to a plant. One plant will have the 17 essential elements. Apart from these, there are certain elements which will help them or they will give a extra help. Extra help dia bostu bilakok ami ki boli koiso beneficial elements boli koiso. For example, we have talked about Na, sodium, right? Na is a beneficial element in case of which type of a plant? In case of halophytes. Halophytes bilakot to ami janu saline area grow kore, right? So tat ki thakibo? Sodium thakibo, and it helps in C4 pathway. Keneke help korise tarpor question aha nai. In which way they are helping? Etu question ketiao nahe, how is Na helping the halophytes? The question might be asked is identify the beneficial element. So identify the beneficial element is Na in case of halophyte. Sik teneke kisuman examples ase Si in case of grasses, cobalt. Yeah, yate bhalke likha nai. Yes, in case of leguminous plants, right? So hekini sam ami eta SI aru eta SE, no? Astragalus SE, Selenium, aru etu leguminous plants at Cobaltia, root nodule formation. Next, toxicity dia ase, toxicity concept to ki jiman lage tatke jodi beshi ke thake tak ami ki boli kom toxic. Most of the micronutrients become toxic. Toxicity of Mn, that is the only example that you have to read from this section. Aru eku na lage, right? Toxicity of Mn. So Mn or jodi toxicity hoy, if a plant suffers from toxicity of Mn, tetia hole kisuman deficiency hobo. Kihor deficiency hobo soason likha ase. Toxicity of Mn induces deficiency of iron, Mg and Ca, right? Eto direct question ahe je Mn or toxicity car deficiency cause kore, right? So hekini important boli likhi thoi dia. Mn toxicity causes crickle leaf disease. Again, this is one very important disease. Why? Because if you remember, when we were talking about bacteria, we were talking about fungi or viruses, we have talked about different types of plant diseases. Fungal disease, viral disease, right? Bacterial disease. So this is also one disease of a plant that is not caused by microbes or pathogens, but instead it is caused due to toxicity. So hekarne hetu question ahibo pare, crickle leaf disease is caused due to, option thakibo due to bacteria, option thakibo due to virus, kintu answer to ki hobo due to toxicity of Mn, right? So Mn toxicity matra eta disease e porim, that is crickle leaf disease. A leaf to crickle kiyo hoy karon deficiency of calcium. Crickle mane horu hoi jay, no, squeeze hoi jay. Okay. Next, toxicity holds a micronutrients bilak. Normally toxic kune kore micronutrients bilak. Jiman quantity plant dalot thakibo lage tatke jodi okon beshi ke thake tak ami ki boli kom toxic. Jiman micronutrients jiman thakibo lage tatke jodi kom ke thake tak ami ki boli kom deficiency, right? Next is mobility of elements, important again. What are the mobile elements? What are the immobile elements, right? Mobile elements kita mukhosto koribo lagibo NPK, Cl aru Mg. Whenever we talk about these mobile elements, deficiency symptoms first appear in older parts only or older plant parts. So question ki hude je nitrogen deficiency hole symptom to ami kot dekha pam, younger part or dekha pam ne older part or dekha pam? Okay, right? Okay, so the question that you will go through is like this. So we have certain mobile elements and we have certain immobile elements, right? So question 2 keneke ahibo, what is the point that is important? First, identify the mobile elements, identify the immobile elements. So mukhosto koribo lagibo. Second, the second statement, mobile elements or jodi deficiency hoy, symptoms ami ketia kot dekha pam? Older parts of a plant. As the immobile elements or deficiency hoy, tetiale first symptoms ami kot dekha pam? Young plant. So a duta statement if you have to remember, so that if the question is asked deficiency of iron, a plant is suffering or a plant is showing iron deficiency. Symptoms of the disease first will appear in the older plant part ne younger plant part. Jehetu নহয় এটো তোমার আয়রন কি হয় immobile, যদি immobile হয় first symptoms আমি কো দেখা পাম younger part, right? If suppose we are talking about calcium, if you are talking about magnesium, these are mobile elements, তাহাতৰ deficiency first আমি কো দেখা পাম old plant parts বিলাকত, আগতে হোৱা plant part নতুনকে বাদ এটা ফর্ম কৰিছে তাত আমি দেখা পাম নেকি নাপাও, তাত আমি দেখা নাপাও. Is the point understood, right? So mobility of elements হেতু paragraph to important বুজি পোৱা নে? Okay, line to পঢ়া সো, line to পঢ়া. First point কি কৈছো, কিছুমান elements ক আমি মোবাইল কও. কিহৰ কাৰণে মোবাইল কও? যে প্ল্যান্টৰ যোনটো পাৰ্টত সিনথেসাইজ হৈছে সেই পাৰ্টটোৰ পৰা বেলেগ এটা পাৰ্টলৈ হি মুভ কৰিব পাৰে, ৰাইট? এটা এৰিয়া সাপোজ এটা নোড আছে, নোডত এটা লিফ আছে, ঠিক আছে? লিফটো সৰি গ'ল, সৰি যোৱাৰ পিছত সেই নোডটোত কিছুমান এলিমেন্টছ আছে, সেই এলিমেন্টছ খিনি লিফটোক দি আছিলে নিউট্ৰিয়েন্টছ, লিফটো যেতিয়া সৰি গ'ল elements খিনি তাতে থাকি গ'ল he can move from that place to the other place and help someone else that is called as mobile whereas on the other hand there are certain elements jot form কৰে তাতেই থাকে তাৰ পৰা লৰচৰ নকৰে ঠিক আছে হি কাকো বেলেগক হেল্প কৰিব নোৱাৰে হি জতে আছে তাতে থাকিব তাক আমি ইমোবাইল বুলি কৈছো ওকে নাও কেটামানক মোবাইল কৈছো কেটামানক ইমোবাইল কৈছো কিয় কৈছো we understood next is যদি মোবাইল এলিমেন্টৰ ডেফিচিয়েন্সি হয় এডাল প্ল্যান্টৰ ডেফিচিয়েন্সি হলে কি হ'ব প্ল্যান্টডালৰ যিকোনো এটা এলিমেন্টৰ যদি ডেফিচিয়েন্সি হয় সাপোজ আ প্ল্যান্ট ইজ সাফারিং ফ্রম নাইট্রোজেন ডেফিচিয়েন্সি হাউ উইল ইউ নো কেনে গম পাবা কিবা কিবি সিম্পটমস হ'ব নহয় ৰাইট সেই সিম্পটমস বিলাক আমি কোড দেখা পাম younger plant parts or the older plant parts or if you are talking about a plant gutekhini part edinote to form kora nai hoy noy ta eta leaf young leaf ase old leaf ase prothomote symptom to jodi old leaf or dekha pua teta we know we are talking about a deficiency of mobile element kine ekdom notun ke kuhi pat eta jodi tumi symptoms kini dekha pua so younger leaf hole nai symptoms to sob gutei gos dalot sobotse beya ke effect kot hoise young parts bilak তেতিয়াহলে আমি গম পাম যে এটো ইমোবাইল এলিমেন্টছৰ কাম যদি ওল্ড প্ল্যান্ট পাৰ্টছৰ হৈছে তেতিয়াহলে মোবাইল এলিমেন্টছৰ কাম বুজি পাইছে ৰাইট সো হোৱাট ইউ হ্যাভ টু ডু ইজ নাথিং বাট বাই হাৰ্টে কৰিব লাগিব ইয়াত বুজিব লগা একো নাই ৰাইট আৰু বেলেগ কোৱেশ্চন একো নাহেও সো ইমোবাইল এলিমেন্টছ causes or shows the symptoms in young parts আৰু mobile elements show the symptoms in older plant parts what are the mobile elements what are the immobile elements Next, eta word dia ase hydroponics. What is hydroponics? Just etai line ahe. Soilless culture is called as hydroponics. What do you do in hydroponics? Culture to code koru liquid medium right? Soil or jimanbur nutrients ase. Edal plant e soil or pra jimanbur nutrients anibo. Gutei nutrients if it's present in the form of water and you can give it to the plant. What will be the necessity of a soil? The plant doesn't need the soil then. Right? So, where in certain places or in certain conditions where there is a scarcity of soil, ami cultivation keneke koribo paru, we can go for liquid cultivation that is called as hydroponics. And what is hydroponics? We'll have a large tank that ami ki korim, we'll give the nutrients in liquid form, right? We can maintain everything, we can maintain the pH, we can maintain the air, etc., etc. So, artificial cultivation in a liquid medium in a specific tank that is called as what? Hydroponics. Soil or plant ki loy nutrients kini moi jodi plant ok beleg eta through diu tete ki hobo eketai kotha nohol niki soil hot pani ase nutrients ase panir logot ekelege nutrients bilak plant dalor tat goise nai goise moi soil tu gusai disu eta bottle thoi disu bottle tu hei nutrients kini e thoi disu eketai kotha hol ne nai soil or pra loise ba panir pra loise it's the same thing right so soilless cultivation buli koy so soil je lagiboi cultivate koribole not necessary soil or pra jikini nutrients no ekini no ekin to ami artificially korisu no you should know entirely about the process then only you can go for hydroponics all of a sudden plant eta uthai dila pani thumai dila teneke nohobo to right you have to be an expert for that right okay next hekini ase introduction part next is nitrogen nutrition whenever we talk about nitrogen nutrition we have to talk about the nitrogen cycle to diagram to dia ase nitrogen cycle boli kwa loge this is something that we have also drawn in I think the chapter organism and population koraisilu niki sulfur cycle carbon cycle nitrogen cycle koraisu tate okay anyway so look into the cycle of question ahibo pare ecosystem ot korisu na okay is the cycle understood atmosphere par nitrogen kole ahibo lagibo soil ole that process is called as nitrogen fixation mixing of atmospheric nitrogen to the soil is called as nitrogen fixation nitrogen fix keneke hobo pare either it could be physical or it could be biological hobo lagile tat ki lagibo microorganisms lagibo if it is physical it could be industrial it could be electrical also thunderstorms hole ki boli koy etu lightning matit pore je right? Thunderstorms hol tat ki hoise actually gas bilak mix howa nai jano, mix hoi pela tolot aha nai jano. So ultimately what is the process that's going on? Some amount of atmospheric nitrogen has been added to the soil. Nitrogen atmosphere pra matit mix howa to ki ami ki kau nitrogen fixation. Keneke mix hoise? Lightning or karne hoise. That's a part of physical, right? Fertilizers disu. আমি এডাল প্ল্যান্টত ফার্টিলাইজার্স দিও ফার্টিলাইজার্স বুলি কলে ম্যাক্সিমাম মোস্ট ইম্পর্টেন্ট কম্পোনেন্ট কি নাইট্রোজেন right so that is also mixing to the soil that is also nitrogen fixation but that is a physical nitrogen fixation বা artificial nitrogen fixation nitrogen fixation biologically অকল যে microorganisms এ atmosphere পৰা nitrogen to আনি পেলাই soilত mix কৰিব not necessary no আমি তো ফার্টিলাইজারও দি দিব পাৰো ফার্টিলাইজার দিলে হেটো আমি কি বুলি কম physical আর যদি rhizobium বা microorganisms involved হৈ আছে তাক আমি কি বুলি কম biological right after the nitrogen is mixed in the soil the plants will take in that is called as ki uptake right plants or pra kot jabo animals tar pisot ki hobo both of them will get decomposed after death after decomposition the amount of nitrogen will go back to the atmosphere that is called as ki denitrification. So after the cycle junkhini dia ase details of dia ase eneke questions ki to aha nai thik ase etu question aha nai je discuss or what will happen or how does nitrogen gets fixed because of lightning teneke question aha nai kintu moi yate details or di disu if you see the reactions swasun physical nitrogen fixation ot ki likha ase two types industrial and electrical approximately 10% of nitrogen is fixed by physical process আৰু steps খিনি দেখাই দিছো যে physical process বা lightning on কি হয় you have nitrogen you have oxygen in the atmosphere both of them will combine and ultimately they will form ki nitric oxide plus oxygen form korise ki NO2 ki hoise along with rainwater falls to the ground and then they will form ki HNO2 aru HNO3 tar pisot ki hobo ultimately everything will get converted to or maximum amount would get converted to nitrates and the plants will take in. All microorganisms say je atmosphere or nitrogen to nitrate or convert korise not necessary. Physical process or karoneo atmosphere or nitrogen to soil or nitrate form or convert hobo pare. Hey prostutue ami dekhaisu. Physical nitrogen fixation or kiba confusion আছে নেকি সোয়া ইমান বেছি ইম্পৰ্টেন্ট নহয় বাই হাৰ্ট নকৰিবা
ডায়াগ্রাম আছে দেখা যায় ডাইরেক্ট অ্যাটমসফিয়ার নাইট্রোজেন ডাইরেক্ট মানে সয়েল যে আহে এটু বায়োলজিক্যাল নাইট্রোজেন ফিক্সেশন মানে লিখি দিছে
biological nitrogen fixation যদি microorganisms helps in the fixation কিন্তু ফার্টিলাইজার যদি দিছো হেতু তো মাইক্রোঅর্গানিজমে দিয়া নাই ন হেতু ফিজিক্যাল যদি রাইজোবিয়ামৰ আমি নাইট্ৰোজেন ফিক্সেশনটো কথা কওঁ তেতিয়ালে হেতু বায়োলজিক্যাল ঠিক আছে ফিজিক্যাল পাৰ্টৰ কিবা কোৱেশ্চন আছিল নেকি not very important what is important is biological nitrogen fixation coming to biological nitrogen fixation swasun there are two types symbiotic or asymbiotic. Please go to the next page. The points that are given below hegita koman important right free living aerobic nitrogen fixing bacteria free living anaerobic nitrogen fixing so aerobic word 2 anaerobic word 2 hegita monot rakhiba question ki ahibo identify the free living aerobic nitrogen fixer identify the free living anaerobic nitrogen fixer identify the symbiotic nitrogen fixer as identify the root nodule formation of helping nitrogen fixer so yare examples kini ami poribo laibo under which heading aerobic anaerobic photosynthetic chemosynthetic hekini part pori swa please read that page and see whether you have understood or not what you have to do important likhi thoba I think it's already written in the PDF right side of important bul likhai ase free living aerobic nitrogen fixing Bacteria question has been asked about Azotobacter and Beijeringia. এই দুইটাৰ ওপৰত বিৰাট question আহিছে free living anaerobic Clostridium ওপৰত question আহিছে আৰু Bacillus এই চাৰিটা example মনত ৰাখিব লাগিব So nitrogen fixation মানে কি atmosphere or nitrogen to soil ot mili joa process to ke ami ki boli ko nitrogen fixation boli ko so atmosphere's nitrogen will get mixed to the soil by which method either biological or it could be physical if you are talking about biological mane ki we are talking about the microorganisms so these microorganisms would be responsible for fixing the atmospheric nitrogen to the soil next whenever we are talking about microorganisms these microorganisms could either be symbiotic or it could be asymbiotic. If they are symbiotic, then they will stay inside the plant body. If they are not symbiotic or asymbiotic, these microorganisms will stay freely in the soil. Is the point understood? Symbiotic, asymbiotic bujisu. Symbiotic hole plant dalor bhitor thakibo. Asymbiotic hole matit thakibo. Right? Aru symbiotic jodi hoy nitrogen fixation or product to code form koribo nitrate code form koribo nitrogen fixation mane ki nitrogen code ase atmosphere ot kole ahibo soil ot ahi ei nitrogen to ki form koribo NO3 negative kune korile since we are talking about biological microorganisms bilak korile microorganisms তাহাঁতৰ কাম কি যে নাইট্ৰজেন টু তাহাঁতে কি হলে কনভার্ট কৰিলে NO3 লে microorganisms are of two types symbiotic asymbiotic if it is symbiotic this process occurs inside the plant right if they are asymbiotic this process occurs in nitrate code form koribo asymbiotic soil symbiotic nitrate code form koribo because that microorganism itself is inside the plant body you have read about this no rhizobium and the root nodules or rhizobium and leguminous plants rhizobium to ki hoy leguminous plant or root or bhitor di khumai jay na jay so plantor bhitor thake so plantor bhitor jodi rhizobium to ase nitrate to code form koribo plantor bhitor right so that is symbiotic if it is asymbiotic nitrates would be formed in the soil right symbiotic biological nitrogen fixation or we have certain examples it is not always rhizobium that is the sole example kitu aru examples of BGA no BGA ami kiman examples koriso one of the most important function of BGA is fixation of atmospheric nitrogen for example ki nostoc Anaerobic olosira etc right ekhini examples agote porisu corelloid root I think you remember we have studied that right why does the root become corelloid or corel like because of the symbiotic association right cycas has two types of roots the normal root and also the corelloid root some parts of the root becomes corelloid or corel like because of what because of the symbiotic association right symbiotic association of whom Anaerobic right nostoc Anaerobic yeah Next, after nitrogen fixation, we have something called as nitrogen metabolism, right? Role of nitrogen in plants. We have been talking about the plants taking in nitrogen. The first point is, why do the plants need nitrogen? Plants need nitrogen because nitrogen is a chief constituent of many elements, right? If you are talking about ATP, if you are talking about GTP, as juntu likha ase protein synthesis karone nitrogen lage বহুত ইম্পৰ্টেণ্ট কামবিলাক কৰিব লাগিলে প্লান্টছ বিলাকক নাইট্ৰজেন লাগে আৰু সেই কাৰণে তাহাতে এটমস্ফিয়াৰৰ পৰা নাইট্ৰজেন লয় ৰাইট ওকে
এটো এই সিম্বায়োটিক যেটো মানে নাইট্ৰেট টু মানে প্লান্টৰ বাহিৰত ফৰ্ম কৰিলে প্লান্টৰ ভিতৰত লাগি থাকে
along with the water nutrients বিলাক কেনেকৈ যাব along with water is a so soil already nitrates ase tahante pani plant absorb koribo roots se yate absorb koriboi na lage karon bacteria to already bhitorte khumai dise right? Okay, nitrogen is necessary for to plants for etc., reproduction, growth, metabolism, etc. That is the reason why nitrogen needs to get fixed. The next point is plants bilake nitrogen korpora pai. Is it only the soil that they get the nitrogen, or there are different sources also? There are different sources also. Khali se soil or prai pabo lagibo in the form of nitrate, not necessary. I think you all have heard something called as insectivorous plants also. Insectivorous plants bilake insect kyo karone khai? Why do the plants take in the insects or eat the insects? Because of nutrition. Why? Ami chicken khwa nisanike. We want to have chicken, so we are having it. They want to have insects, so they are having it. Tenika niki nohoy. Why they are having that? To what? Make up their nitrogen deficiency. They take the insects, in the insect's body ki thakibo nitrogen, that is the nitrogen that the plants use. Insect opra nitrogen kyo lobo laga hol tahator? Because they grow in nitrogen deficient soil. Junbilak plants e juntu soilot nitrogen ase, soilor prai to direct loi lobo, right? Jedi soilot nitrogen e nai, plant dale nitrogen korpra lobo. They will take it from those insects body. Insects or body nitrogen thakibo tahate loi lebo. So why do the plants eat the insects? To make up for their nitrogen deficiency. Why? Because they grow in a nitrogen deficient soil. So what we can conclude, it is not only from the soil that they get nitrogen, they can get nitrogen from different sources also, right? So what are the sources? Likha ase, plants obtain nitrogen from various sources. First, the most common, that is atmosphere. From the soil in the form of NO2, NO3, eketai point hetu. Next, organic nitrogen in the soil, third point. Soilot nitrogen duta formot thake, eta hol organic, eta hol inorganic, right? If you talk about inorganic form of nitrogen, we are talking about what? Ammonia, nitrites and nitrates, right? If you are talking about organic form of nitrogen present in the soil, we are talking about the process of decomposition. Edal plant jeta decompose hoy, tar nutrients bilak goi pelai soilot mix hoy nohoy. Right? So, if a plant or an animal gets decomposed, the constituent that was present inside the body of the organism mixes with the soil, and that part would be called as what? Organic part, right? Hey, organic part of nitrogen asile. So, ultimately soil two types of nitrogen thake, organic nitrogen, inorganic nitrogen. Inorganic nitrogen microorganisms bilake directly atmospheric nitrogen to convert korile ki hole ammonia, nitrites or nitrates hole. Aru organic nitrogen source ki the decomposition process, right? Tar likha ase, so aposto to buzi paisa nai. The fourth source is insect body. Iman details or nahe de. Yar bitor moi jungita important point koisu hegirai porhim. Nitrogen fixation porhim, examples porhim. Thik ase iman details na lage. And the
asymbiotic mane biological nitrogen fixation
kun kita?
Mane asymbiotic biological
egita question ahe. Special number 1, number 2 to birate question ahe. Azotobacter, Basilinkia, Clostridium, Bacillus. Esa ita birat question ahe. Because nitrogen fixation, whenever we hear, we have a tendency to think that it is always symbiotic, but we know it is not always symbiotic. It is asymbiotic also. Ekane asymbiotic or examples kini upor question ahe. Next is nitrogen in the form of urea also. Plants bilake okol je soil opra pabo, not necessary, they get it in the form of urea or fertilizers. Next point talks about one word that is called as diazotroph or diazotrophy. Ei duta word janibo lagibo. What is diazotroph and what is diazotrophy, right? So nitrogen fixing organisms are also called as diazotroph. Junbilake nitrogen fix kore, hey organism, Rhizobium, nostoc, Anaerobic, these are also considered as ki diazotroph. Our nitrogen fixation process took ami aru eta naam ase, he naam to ki diazotrophy, right? So what is diazotroph and what is diazotrophy? Next, chart খন চোৱা. Diazotrophs or the microorganisms that fixes nitrogen could be free living or symbiotic. Free living, we have already discussed the examples, Clostridium and all those. অলপ আগতে দিছিলোঁ নাই যোনবিলাক পইণ্টছ আছিলে, ৰাইট? ফ্ৰী লিভিং নাইট্ৰজেন ফিক্সাৰ্ছ এক্সাম্পলছ অলৰেডি দি দিছোঁ. এইখন চাৰ্টত কি দিয়া আছে? চিম্বায়টিক নাইট্ৰজেন ফিক্সাৰ্ছ. আণ্ডাৰষ্টুড অৰ ডিড নট আণ্ডাৰষ্টেণ্ড? Actually, I have given shared the PDF so many days before. তোমালোকে কি এবাৰো পঢ়া নাই নেকি? নে ডাইৰেক্টলি ক্লাছত লৈ আনিছা? What is the point of sharing the PDF 1 week before the class? So, whenever you get a PDF, whenever you get a note, at least ঘৰত এবাৰ পঢ়া ন? হয় নহয়? নপঢ়িলে কেনেকৈ হ'ব? So, পঢ়ি আহিলে কি হ'ব? You'll know the sequence. এতিয়া তোমালোকৰ প্ৰব্লেম হৈছে. You are not being able to like catch it very rapidly or fast. Fast ল'ব পৰা নাই. মই কি কৈ আছোঁ তোমালোকে পইণ্টছৰ মাজত, পেজৰ মাজত কনফিউজ হৈ গৈছা. What I am saying is nitrogen fixation. Nitrogen fixation বুলি ক'লে the first word that comes to our mind is symbiotic nitrogen fixation. Is it always symbiotic? No, it is asymbiotic also. Examples of certain microorganisms who fixes nitrogen asymbiotically. কি example হ'ব? Is a aerobic, anaerobic, photosynthetic, chemosynthetic বিলাক ক'লো, azotobacter ক'লো ন, Beijeringia ক'লো, Clostridium ক'লো, Bacillus ক'লো, Nostoc, sorry, এইকেইটা স্পেচিয়েলি কি হয়? Asymbiotic. Asymbiotic বুলি ক'লে the best example to Nostoc আছেই, right? Okay. Again, whenever we talk about symbiotic nitrogen fixation, আমাৰ দুটামান ৱৰ্ড সদায় মনত থাকে. এটা হ'ল নাইট্ৰজেন ফিক্সেশ্যন, এটা হ'ল চিম্বায়টিক নাইট্ৰজেন ফিক্সেশ্যন, এটা হ'ল ৰুট নডিউলছ, এটা হ'ল লেগুমিনাছ প্লান্টছ. নাইট্ৰজেন ফিক্সেশ্যন বুলি ক'লে এই চাৰিটাই জানো, ৰাইট? বাট দিছ চাৰ্ট টেলছ আছ অকল যে নডিউলছ ৰুটতে ফৰ্ম কৰিব, নট নেচেচাৰী. ইন চাৰ্টেন প্লান্টছ দ্য নডিউলছ আৰ ফৰ্মড ইন দ্য ষ্টেম. ইন চাৰ্টেন প্লান্টছ দ্য নডিউলছ আৰ ফৰ্মড ইন দ্য লিফ. But what we will study in details is root nodule formation. That does not mean nodules are always formed in the root. Kisuman plants are rootot nodules to form koribo pare. Kisuman plants are stemot form koribo. Kisuman plants are leafot form koribo. Right? So this chart just tells us the nodule code form kore. Nodules are called rootote form kore, not necessary. GT part of nodule form koribo pare. Next. Symbiotic nitrogen fixation hole, is it necessary that every time nodules will be formed? Please listen to the question. Symbiotic, is it necessary that whenever we are talking about symbiotic nitrogen fixation, formation of nodule is a must? Is it a must or it's not a must? It's not a must. If we are talking about Rhizobium, if we are talking about leguminous plants, then nodule formation is a must. If you are talking about fixation by blue-green algae, nodule does not get formed. So, nitrogen fixation nodule form koribo pare, form no koribo pare. That is the information that is given in the chart, right? The points are important. Points kita bhalke poribo leibo, right? Again, so nitrogen fixation ki kolu? Symbiotic, asymbiotic. Symbiotic, no nodules or nodules, right? Whenever we talk about nodules, we talk about ki leguminous plants, right? Kintu there are certain non-leguminous plants also where the nodules are formed. Just turn the next page. Root nodule forming with leguminous plant, root nodule forming with non-leguminous plants. Parisa identify koribo. Out of these 2 points, which one is the common one? Tumalke kuntu jana? Leguminous plant. So question leguminous plant or nahe. Question tumalke juntu nazana hetu upor ahibo. Right? Root nodule formation in non-legumes. That is the most important question that is asked. Right? So leguminous plants are to ami janu any gram pea these ebla ki hoy leguminous plants that ki form koribo rhizobium e root nodules form koribo. So that is such a common point that we know he karone tar upor question nahe. Question to ki ahibo যে root nodule non legumes ot form kore nokore, obviously kore, right? And what is the, this is something that we have read before also, Frankia aru ulnus. Frankia ulnus hetu hol ki non legumes plants ot root nodule form koribo. Next, root nodule in non leguminous plant ulnus is formed by bacteria Frankia. Is that point understood? Point buji paisa nai? Paisa? And that point is important. So this was all about nitrogen fixation. So what we have understood when we talk about nitrogen fixation, mixing of atmospheric nitrogen to the soil is called as nitrogen fixation. How does the process takes place? The process takes place either physically or biologically. Which part you have to read in details? Biological. Whenever you talk about biological nitrogen fixation, it could be symbiotic or asymbiotic. Examples of symbiotic nitrogen fixers and examples of non-symbiotic nitrogen fixers. Next, if you are talking about symbiotic, we talk about root nodule formation in legumes, but root nodules are also formed in non-legumes, right? If you are talking about legumes, which bacteria is responsible? Rhizobium. If you are talking about non-legumes, which bacteria is responsible? Frankia, right? Likhat bahut ase, but the points egitai poribo lagibo. Next, we go back to that nitrogen cycle, and we have a process called as ammonification. What is ammonification? The process of formation of ammonia is called as ammonification, because we know atmosphere ot ki asile nitrogen, soil ot ki form koribo, directly nitrate form koribo ne prothomote ammonia form koribo. First ammonia, right? So if you are talking about nitrogen in the atmosphere, first ki form koribo NH3 NH4 zilika ammonia. Ammonia ki hole convert hobo, tapsot ki hole convert hobo, right? A process tok ami ki boli kom ammonification. The process of formation of conversion of atmospheric nitrogen to the form of ammonia is called as ammonification. Who is responsible? As a ammonia form korile nitrogen ekune korile, certain ammonifying bacteria, right? So ammonifying bacteria examples, example bacillus, right? So ammonification or ki porhim bacillus mycoides and bacillus ramosus. Question keneke ahibo? Identify the ammonifying bacteria. Right? So ammonifiing bacteria. What is ammonification? Question nahe. Examples to upor question ahibo. Right? Next is nitrification. A2 part ami biological classification of korisu. Right? Again, we have a word called as nitrification. What is nitrification? Conversion of ammonia to nitrite and nitrite to nitrate is called as nitrification. Who is responsible? Groups of bacteria. So identify the nitrifying bacteria. Nitrosomonas, Nitrobacter. Right? Okay, nitrification at a point that is new. So what is nitrification? We know. Which group of microorganisms is responsible? We already studied. Just come to the last point. At a fungi report point dia ase swa. Fungi that helps in the process of nitrification. Duta question hobo pare. Identify the bacteria that helps in nitrification. Bacteria ki hobo? Nitrosomonas, Nitrobacter. Next question ki ahibo pare? Identify the fungi that helps in nitrogen fixation. Aspergillus flavus. Paisa ne last point too, right? So last point too important. Huh? Bhalke aha nai? Aspergillus. Reaction to ase swa? Aspergillus, FLAVUS. Bacteria involved in nitrification, fungi involved in nitrification. So question has been asked about fungi involved in nitrification. Right? Buji paisa ne kun kita important point mark kori diso, right? Okay. So ammonification we understood nitrification also. Microorganisms examples ami porim. The next point is denitrification. What is denitrification? The movement or the process through which soil nitrogen goes back to the atmosphere is called as denitrification. Who is responsible? Again microorganisms. Hey, microorganisms bilakok ki boli kom? Denitrifying bacteria, but denitrifying microorganisms. Examples easy to remember, karan species or denitrificants word to likha ase swa. Pseudomonas denitrificans, Thiobacillus denitrificans. These are certain microorganisms that are considered as key denitrifying bacteria. All. And what is the process? Tolot reaction to diases. Nitrates ki hole convert hobo or ultimately gaseous form what it will go back to the atmosphere. The process is called as ki denitrification. Reaction not important. What is important? Examples. Ekin clear? Up till here what we have studied? We have talked about the macro elements, micro elements. We have talked about essential elements. We have talked about the criteria for essentiality. Next is something called as toxicity. Next, mobile elements, immobile elements. Mobile elements are logot older plant parts, immobile elements are logot younger plant parts. After that, we have discussed the nitrogen cycle. Talking about nitrogen cycle, another point we have talked about nitrogen fixation. Nitrogen fixation ekhini bostu porhilu. Nitrogen cycle or logot process like ammonification. Who is responsible? Ammonifying bacteria. Denitrification, denitrifying bacteria. Nitrification, bacteria is involved. Together with that, a particular fungi is also involved, that is Aspergillus flavus, right? So hekin loke porilu. The next part of the note talks about nitrate reduction. This part says that what will happen after nitrate enters the plant cell. Nitrogen kot ahile soilot in the form of ki nitrate, plante nitrate absorb korile, cell tur bhitorot nitrate or ki hobo. That is what is called as nitrate reduction. Nitrate will get what? Converted to nitrite, nitrite will get converted to ammonia, ammonia will get converted to amino acids, amino acid will get converted to amides. So this is the process. Clear? So nitrate reduction or bhitorot, nitrate reduction, reduction of nitrate occur in two steps. Please come directly to the step. So eta plant cellot etia ki ase NO3 negative, right? This is the plant cell after absorption. A cell tut ki hobo junto NO3 negative ase hi ki hole convert hoy jabo NO2 negative. E ki hole convert hobo ammonia, right? A process to kami ki buli koiso nitrate reduction. Reduction hobo noy NH ahi jabo no yate, yate to H nai no. Nitrate, nitrites, subset ki hobo ammonia ahibo, ammonia hole to H thakibo NH3, NH4. So a process to kami ki buli koilo nitrate reduction kot hoy ase plant cell to bhitorot hoy ase. Nitrate reduction occurs in two steps. First, this is one, nitrate to ammonia 2. Understood? Who is responsible? Certain types of enzymes, certain types of organisms. So nitrate reduction, come to that point. Reduction of nitrate to nitrite is carried out by an enzyme molybdoflavoprotein. A bostu to important. Jeneke ami nitrosomonas group and nitrobacter group. These are the two things that we have studied. Oh no, olop agote ki porisilu je nitrogen ase, nitrogen ki hole convert hobo ammonia le, ammonia ki hole convert hobo N2 or N2 ki hole convert hobo N3. Ekhini kot hoy ase, especially we are talking about in soil. What we have talked about is yate ki asile somonas, yate ki asile nitrobacter. Ekhini amar clear? Etia plant cellor bhitorot cell tur bhitorot ki hoy ase NO3 ki hole jabo NO2 tarpsot ki hole jabo ammonia. Yate eta enzyme lagibo, yate eta enzyme lagibo, right? Prothom enzyme to ki molybdo ta mane ki ase MO ase in the enzyme molybdoflavoprotein. Our next enzyme is olop pisot pam metalloflavoprotein. So enzyme kita monot rakhibo lagibo. Yat jeneke nitrosomonas aru nitrobacter ami monot rakhisu, thik teneke yate a enzyme duta monot rakhibo lagibo. Clear? Come to that step. Carried out by an enzyme molybdoflavoprotein. So question ahe molybdoflavoprotein e kuntu reaction ot help kore? Reduction of nitrate to nitrite inside the plant cell. right? Okay. It also requires a coenzyme. Ekhini iman important nohoy, kintu name of the nitrate reductase is very important. A enzyme to, so please look into this. This enzyme ki korise nitrate ki hole reduce korise NO2. So this enzyme could also be said as nitrate reductase. This enzyme nitrite reductase. Nitrate reductase kun molybdoflavoprotein. Nitrite reductase ki metalloflavoprotein. Clear? Okay. Our reaction not aru kiba kibi use hobo jeneke coenzymes ase, right? Flavin ase, etc. But what is very important from this point is the name of the enzyme. Ami enzyme to monot rakhim, reaction ami monot na rakho karon question aha nai etialake, right? So NO3 with the help of other substances will get converted to nitric inside the plant body. Next step, reduction of nitrites to ammonia. It is carried out by an enzyme that is called as metalloflavoprotein. That is also called as nitrite reductase. Yeah. So ultimately inside the plant body ki ase etia ammonia. Yes or no? Right? So after this conversion, after this reduction, it is present in the form of ammonia. What I said, ammonia with the help of different substances will ultimately form amino acids, right? Are amino acids kune form kore? Amides bilake, right? So the next part talks about biosynthesis of amino acid. Amino acid keneke form korile ammonia pora. Right. So ammonia pora amino acids keneke form korise. First process ki dise reductive amination. Right. By a process called as reductive amination, amino acid has been formed inside the plant body. So what happens in reductive amination? Ammonia was already present. Ammonia kabar logot combine koribo. Kar logot koribo alpha ketogluteric acid. Alpha ketogluteric acid, if you remember, Kot porisila? Citric acid cycle. Respiration of alpha ketogluteric acid, succinyl coenzyme asile, alpha ketogluteric acid asile, right? So in the cell, the process of photosynthesis is going around, the process of respiration is also going on, and various type of activities has been going around in the cell. Because of these processes, different substances are forming inside the cell. So yate jete ammonia form korile because of nitrification, because of nitrate reduction, ammonia ki koribo? Alpha keto was already present in the cell because of citric acid. Both of them will combine and ultimately they will form amino acid. Is the point understood? Cell tut ki asile? Ammonia. Ammonia ki koribo? Kabar logot combine kori amino acid form koribo. Kar logot combine koribo? Alpha ketogluteric acid or logot. Alpha ketogluteric acid cell to bitot keneke ahile? Konobai to form korise no tate. So because of what? Citric acid cycle. Respiration hoy ase citric acid cycle hoy ase, glycolysis hoise, glycolysis apor citric acid cycle hoise. So citric acid cycle or eta byproduct ki alpha keto. In the entire citric acid cycle, obviously you have to by heart that oxaloacetic acid, malic acid, right? Then cis trans acylate upside succinyl coenzyme. So you have to by heart in the entire cycle what is the two very important product, succinyl coenzyme or alpha ketogluteric acid. Right? So alpha ketogluteric acid or logot mili palai tahante ki form koribo? Amino acid form koribo. This process is called as reductive amination. Clear? So alpha keto logot log lagi palai ki form koribo? Glutamic acid. So what is the first form amino acid inside the plant cell? Glutamic acid. Right? Glutamic acid kune form korile? Ammonia plus alpha ketogluteric acid. Right? Sometimes ammonia also gets or also combines with oxaloacetic acid. এমোনিয়াই বেলেগ বেলেগ বস্তুৰ লগত কম্বাইন কৰি বেলেগ বেলেগ এমিনো এসিড ফৰ্ম কৰি থাকিব যদি আলফা কিটোৰ লগত কম্বাইন কৰিছে তাহতে কি ফৰ্ম কৰিব গ্লুটামিক এসিড কেতিয়াবা অক্সালো এসিটিক এসিড আকৌ সাইট্রিক এসিড সাইকেলত পাইছো আমি right so যদি এবাৰ ammonia oxalo acetic acid if it combines with that then they will form another type of amino acid that is called as aspartic acid right so nitrogen asile soil atmosphere nitrogen soilot ki ahile nitrate this nitrate inside the plant cell ultimately formed ki ammonia combined with maybe alpha keto sometimes it might get combined with oxaloacetic acid and because of this ki form korile amino acid alpha keto logot jodi kore glutamic acid oxaloacetic acid aspartic acid out of these two the question has been asked about glutamic acid only right what is the first form amino acid inside the plant body glutamic acid so by a process called as reductive amination amino acids get synthesized inside the plant body right ami ki pori asu je amino acid keneke form korise amino acid keneke form korise by a process called as reductive amination jot ammonia belegor logot combine kori ki form korise amino acid form korise or it could be amino acid could also be formed by another process that is called as transamination এটা প্লান্ট সেলৰ ভিতৰত এমিনো এসিড কেনেকে ফৰ্ম কৰিছে দুটা প্ৰসেসৰে ফৰ্ম কৰিব পাৰে আইদাৰ ৰিডাকটিভ এমিনেশন অৰ ট্ৰান্সএমিনেশন ৰিডাকটিভ এমিনেশন মোস্ট কমন আৰু আমি গম পালো ৰিডাকটিভ এমিনেশনত কি কৰে ইট গেটস কম্বাইন্ড উইথ আলফা কিটো অৰ ইট গেটস কম্বাইন্ড উইথ অক্সালোএসিটিক there is another method through which amino acid is formed that is called as transamination what happens in transamination the glutamic acid that was formed glutamic acid kune form korile reductive amination right glutamic acid that was formed combines with some other acids to form another amino acids that is the reason why I said amino acids are not stable তাতে কাবাৰ লগত ৰিঅ্যাকশন কৰি বেলেগ বেলেগ এমিনো এসিড ফৰ্ম কৰি থাকিব পাৰে প্ৰথম কোনটো এমিনো এসিড ফৰ্ম কৰিলে গ্লুটামিক এসিড এবাৰ গ্লুটামিক এসিড কাৰ লগত কম্বাইন কৰিলে পাইৰুভিক এসিডৰ লগত পাইৰুভিক এসিডৰ লগত কম্বাইন কৰি কি ফৰ্ম কৰিলে সচোন এলিনিন right so ebila combination hoy থাকিব out of these two process we will be talking about first one in common aru hetur uporte question ahise sometimes glutamic acid could also combine with oxaloacetic acid and then what they will form is aspartic acid so cell tur bitorot since amino acid is not stable they can keep on reacting with different products that are present in the cell and forming different products ultimately different types of amino acids will get synthesized in the plant body right what is important the point that is given below duta star dia ase sosun glutamic acid is the first formed amino acid in the plant and can synthesize different amino acids by transamination eta amino acid e beleg amino acid form kori thakibo pare right next we have a process called as catalytic yate ki likha ase amination likha ase no it's not amination amidation hobo lage DATION catalytic amidation catalytic amidation or itate heri to dia nai likha asun jot catalytic amidation likha ase no just write one statement there but likha nai kiba what is catalytic amidation Once the plants take in nitrate from the soil, nitrate undergoes reduction and ultimately ammonia is formed. What will happen to ammonia? Ammonia will combine in different products that are present inside the cell and will form amino acids. Since amino acid is not stable, we need a stable form. What is the stable form we are talking about is amide. Who forms amide? The amino acid itself forms amide with the help of certain enzymes. This process is called as key catalytic amidation. বুঝি পাইছে? So, plant cell tut juntu nitrate asile, nitrate to ki hole form korile ammonia, ammonia ki hole form korile amino acids. Amino acids form kora, this process is called as ki reductive amination. Right? Reductive amination or karone ki form korile different types of amino acid. This amino acids will ultimately form ki amides. What is this process called as? Is the point clear? So, অকমান সাইডত তেনেকে লিখি লবা তেতিয়াহলে বুজি পাবা নহলে সব কনফিউজ হৈ যাবা বস্তুটো right? Is this clear now? Clear? এখন flowchart যদি মনত থাকে বাকিটো definition তোমালকে এনেও পঢ়ি লব পাৰা হয় এই processটোক আমি কি কম Yes? So, I think this will make the concept clear. Our nitrosomonas, nitrobacter logot, you have to remember that fungi also. Aspergillus, flavus, Aspergillus, flavus. Out of this, what is the most frequently asked question? What is the first form amino acid inside the plant? Glutamic acid, right? What is the first form amide inside the plant? Glutamine. Jedi amide thake, MINE থাকিব যদি এমিনো অ্যাসিড থাকে গ্লুটামেট ATE থাকিব Econ chart jeti bhalke bonua ghorot if you write it this properly no what you can do with molybdenoflavoprotein what are the extra that we have talked about FMN FAD right iron asile kobat molybdenum asile we have written there so what you can do comma write the information again comma write the information yate jeneke ami nitrosomonas and aspergillus we have written so at home what you can do molybdenoflavoprotein comma FMN FFD etc right And while reading this chapter, nitrogen fixation part to পঢ়িলে প্ৰথমতে এইটো পঢ়িবা তাৰ পিছত নোটছ লৈ যাবা নহলে সব কনফিউজ হৈ যাবা as it is definition তো নাহে কেতিয়াও definition আহিব নেকি নাহে কনসেপ্ট তো বুজি পালে হৈ গল okay come to that part catalytic amidation or last part of কি লিখা আছে সোয়া something about legumes Okay, this amino acid, conversion of this amino acids into amides has been commonly seen in because nitrogen fixation ko aloge we remember that one only as common nitrogen fixation in leguminous plants. So this process আমি কোট স্টাডি কৰি আছো legumes so this process commonly seen in case of legumes the next point talks about two different types of legumes just go through the PDF last line dekhisane legumes of temperate legumes of tropical right so question ahe what is the amide that you will find in the legumes growing in temperate What is the type of amide that you see in case of legumes growing in tropical? Sentence kita parisane, buji paisane? The legumes of temperate origin like pea, example diya ase nai? Yeah, pea aru clover, tat ki paisu? Aspergine, INE. So we are talking about what? Amide, right? The legumes of tropical area, tat ami ki paisu? Like soybean and cowpea that ki ase urides URIDES so etu hol right etu likhilu asai likhai ase What did I write? Transported to the non-nodulated parts. Tar mane? We are talking about what? Amino acids. We are talking about what? Amides. A amino acids, A amides code form kori ase. In the nodules, what is nodule? The collection of the cells. So, hey nodules are bhitor the ase, but these needs to be transported to the different parts of the plant body also. So, non-nodulated parts bilakot, in case of tropical, this is the amide that goes. In case of temperate, this is the amide that it goes. Right? So, question to kihor upor ahise? Eitur upor. Aspergine aru uriza. Temperate aru tropical. Tahator beleg dhoroner different amino acids form koribo, the process would be different. Right? Kintu ami jihetu commonly eitu poru, so we are talking about it in details. Clear? Okay, come to the next part. Ekhon likhila sobe? Likhisa? Ekhon je nilikha confused hoise ba okman likha tete. What about the online students? Have you written this? Okay, please write it properly. Okay, I did not write it very clearly. Mo okman haphazard ke likhisu. The sequence is okay. Maybe the handwriting is not good. Okon bhalke likhiba, bhalke poriba, tar pisot notes poriba, nahole confuse hoba. If this is over, I think I can rub. Okay. Next, we go to the next part of the PDF. Next part of the heading ase swason, symbiotic nitrogen fixation in leguminous plants. Right? Please come to that. Tatu important dutaman point poribo lagibo. Symbiotic nitrogen fixation in leguminous plants. What does that topic talk about? If you are talking about leguminous plants, suppose, obviously soilot ki thakibo, microorganisms thakibo. If this is a root, they will have the root hairs, right? And we know, for example, Rhizobium. Why Rhizobium? Because we are talking about symbiotic nitrogen fixation. That does not mean soilot okol Rhizobium thakibo. Soilot to bahut microorganisms thake. Free form or symbiotic. We are taking the example of symbiotic, so we are talking about Rhizobium. In this soil, there are different plants, not only leguminous. Edal ki hoy? Legume. Non-legumes so thakibo pare, different plants so thakibo pare. Okay. Since we are talking about symbiotic, what we know, this microorganism needs to enter here, right? So if this needs to enter here, then this will have to identify this plant, right? Bahut beleg plants ase. Legume kundal hoy keneke identify koribo, right? It needs to identify. So certain types of chemicals are synthesized. As a result, enters the root or goes near to the root. What happens once the rhizobium reaches the root hair? Root hair or ki hoy? Curling, right? The root was like this, root hairs were like this, right? Once the rhizobium goes nearby, it curls because it has to entrap that rhizobium, right? That's the first step. Second, after curling, this microorganism lyses the cell wall of the root hair. Right? Certain types of chemicals it sensitizes, it lyses and the cell wall at one part, and it ultimately enters the root hair, right? First keneke hol kisman chemicals dile, chemicals of rhizobium to attract hol like curling of the root hair, lysis of the cell wall, entry. Once the bacteria enters the root hair, what it will do? It will multiply and it will cause infection. Obviously, if the pathogen enters the body, ki koribo? Infection cause koribo. So what does this rhizobium do? Will grow and regrow, right? And they will keep on increasing and forming something called as Clear? Rhizobium enter inside the cell, increase its number and cause infection. This is what is called as infection thread. These infection thread will keep on branching and rebranching. Mane the number of microorganisms will keep on increasing, right? And ultimately from the root hair it will enter the first cell. What is the first cell? Epidermis, right? Jedi etu he no, etu epidermis. Next is ECEP pahorila niki? Cortex, pericycle and xylem phloem. Root hair kot thakibo? Because it's epidermal in origin. So if the bacteria enters the root hair, so it will divide and redivide, it will form infection threads. Ultimately it will enter what? Epidermis. Clear? Epidermis otu ki koribo? It will further grow. Infection threads bari goi thakibo, right? After entering, after capturing this, it penetrates the cortex. Once it reaches the cortex, the bacteria does not divide anymore. Thik ase? Etia this bacteria takes up the mechanism of this cell. It stops dividing, and at that time it is called as bacteroid. Bacteroid ketia kom? Jetia non-dividing hobo. When it does not form any more infectious thread, right? So it will become bacteroid. What it will do, if this is a bacteroid, bacteria asile, bacteria ketia korse jetia infection cause kori asile. Jetia infection aru cause kora nai, yate aru divide kora nai, we use a term that is called as bacteroid. What will the bacteroid do? The bacteroid takes into control the machinery of the cortex and allows the cortex to de-differentiate. What is de-differentiate? Bacteria to cortex ot humai pelai aru divide no kore, tetia bacteria to kami ki boli ko bacteroid. First point. Second, bacteroid to a cortex of ki koribo? De-differentiates the cortex. What is de-differentiates? Cortex was a permanent cell. That permanent cell dividing again, the process is called as what? De-differentiation. So this bacteria will allow this cortex to form many cells. This collection of cells, nodules. Who forms the nodules? It is the cortical cell of the root that forms the nodules. What comprise of a nodule? The different collection of cortical cells comprise of a nodule. Inside each and every nodule ki thakibo? Bacteroid. Is the concept clear? Bacteria humale ahile, epidermis cross korile, cortex ahile. Cortex ot ki korile? Cortex took de-differentiate kori dile. It forms a collection of cells. Cortex e ki form korile? Nodules. Inside each and every nodule, what you will see is the bacteroid. Ekini clear? Next part. If this is a nodule, if this is a bacteroid, right? What does the nodule do? The host, etu kami host boli kobo parim ne nokori, right? This is also called as ki? Nodule. Who formed? Cortex. So this is the guest. Once the bacteroid enters inside a nodule, what does the nodule do? The nodule will cover itself with a membrane. আমি যেনেকে আমাৰ ঘৰত মানুহ গলে কি কৰো? খাবলে দিও, গেষ্টক কি কৰো? খাবলে দিও, বহুত দিন থাকিলে কাপোৰ কানিও দিও, হয় নহয়? ঠিক তেনেকে when the guest is inside the house, what they will do? They will give them a cloth. Who synthesizes the peribacteroid membrane? It is the cortex or it is the nodule. What is the reason behind? Kiho karone tak trap kori dile so that it does not go away. If the bacteria goes away, tetihale fixation no hobo, ki nitrite na pabo. So in order to keep it, it entraps in the form of a membrane. Who synthesizes this membrane? The host. The name of the membrane is called as peribacteroid membrane. So once peribacteroid membrane has been synthesized, the bacteroid remains captured inside the cell. First, hoste duta kam kore. Eta to kapur ekhon dilei tar uporot It will synthesize up pinkish liquid that is called as leg hemoglobin. Right? So process to discuss koribole to ketiao nahe khali words bilak janibo laibo. Question to jodi ahe who forms or who synthesizes LB. LB is leg hemoglobin. What is the color? Pinkish, right? Perivectoid membrane kon kune form korile? Host to who is the host? The cortical cells, right? Okay. Now this is a liquid. Ekon kiyo karone form korile? To trap the bacteria. A liquid to kiyo karone synthesize korile? What was the necessity of forming leg hemoglobin? To cut off the oxygen supply. Why? Because nitrogen fixation always occurs in an anaerobic condition, right? So a bacteria to ki korile in and around at a liquid or thoidle so that the medium becomes anaerobic and now he can go for nitrogen fixation or conversion of nitrogen into nitrates inside the plant body. Is the point clear? So this is the process of symbiotic nitrogen fixation. Now read that part. Jaldi porha, we'll finish up this lesson. Symbiotic nitrogen fixation of point key important. Bacteria attracts the rhizobium with the help of certain attractants. What are the name of the attractants? Flavonoids or betaines. Likha ase? Right? So flavonoid, so this root will synthesize what? Flavonoids and betaines. Because of these chemicals, this will get attracted here. Okay. Second, root curling and all those are process to porisa. Moi juntu bostu bujaisu, he bostu to buji paisa nai. Just see, are the sentences going along with the description? I mean, the point that we have explained. Chemical genes, hormones. Read up till nitrogen, read up till leg hemoglobin. Synthesize কৰিছে হস্তে দুটা কাম membrane আৰু liquid membrane খন কিয় কৰে trap কৰি ৰাখিবলে liquid টো কিয় কৰে anaerobic condition কৰিবলে হা oh nodule is the host itself the nodule synthesizes leg hemoglobin right is it understood clear Why anaerobic condition is important or necessary? Because nitrogen fixation occurs in anaerobic condition. Actually, what happens in nitrogen fixation? Atmosphere ki asile? N2, right? N2 will get converted to what? NO3, right? So, a oxygen to log lagibo lagile, N2 to keneke asile? Like this. So, this bond needs to get broken. Then only it will become free and it will take in the oxygen, right? A bond to kune break kore nitrogenase. What is nitrogenase? The enzyme of key microorganisms. That is the reason why microorganisms can fix atmospheric nitrogen directly. Nitrogen or majot bond to bhangibo lagibo jedi bond to bhange tetiahe oxygen accept koribo bond to kune bhangibo only a particular enzyme which enzyme nitrogenase where will you find inside the plant body no If nitrogenase was present inside the plant body, then they could have taken atmospheric nitrogen directly. Since it is not present, where is it present? In the microorganisms. So, plants need those microorganisms to break the bond so that nitrates could be formed. Clear? Our nitrogenase enzyme becomes or deactivated or degenerates in presence of oxygen. That is the reason why anaerobic condition is necessary. Nitrogenase enzyme to oxygen or logot ahar loge loge hi bhangi jabo. A bacteria nitrogen fixation or sobotse important enzyme ki nitrogenase. Hiye jodi bhangi jay nitrogen fixation keneke hobo. So it is very essential that nitrogen fixation always occurs only during anaerobic condition to protect that particular enzyme or to keep that enzyme away from oxygen. Right? So who helps in keeping that enzyme away from oxygen? Hemoglobin. Who synthesizes? The cortex or the nodules. Right? Aerobic hobo লাগিছিল হেকাৰণে এটো পানীত মানুহ এটা পানীত সোমাই দিলে কি হ'ব কিয় মৰি যায় ব্যাকটেৰিয়াটো কিহত সোমাই দিলে পানীত সোমাই দিলে সো তাহাতে অক্সিজেন নাপায় আৰু সেল বাকি সেলটো গুটে অক্সিজেন আছিল কাৰণ ৰেসপিৰেশন হৈ আছে ন অক্সিজেনটো obviously আছিলে right বা photosynthesis হৈ আছে অক্সিজেনটো obviously asile right but since it is trapped by this liquid the condition becomes anaerobic he can oxygen supply to gut. Is the process clear? Clear? Okay. কিয় মনত নাথাকিব এবাৰ ঘৰ ভালকে সেই কাৰণে তো আগতে পঢ়ি আহিব কৈছিলে কিয় পঢ়া নাই হা এবাৰ পঢ়িব লাগে ন ওকে কাম টু দ্য নেক্সট পইণ্ট পইণ্ট দুটামান দিয়া আছে সেই পইণ্ট কিটা ইম্পৰ্টেণ্ট এটা প্ৰটিনৰ নাম দিয়া আছে চোৱাচোন Ricardison protein of bacterial cell helps in identifying the host. মই অলপ আগতে কি কলো যে rootটোৱে কিছুমান chemical synthesize কৰে so that this gets attracted. He also have a certain type of protein that makes it easy to identify the plant easily. So it's a mutual relation. অকল যে plant ডালে chemical synthesize কৰে তেনেকুৱা নহয় ইয়াৰো এটা specific protein আছে Hey protein to karne, he can go closer to the host cell. Right? What is the name of the protein? Ricardison. That is the first point. Second point, nodule formation. Kune korile? Cortex se. By which process? Dedifferentiation. Dedifferentiation kihor karne hoise? It is the action of certain auxins, certain hormones. Cortex se divide and redivide kori pelai. Nodule form korile. Here's a divide and redivide korile. Process is called as what? Dedifferentiation. Why did the cell dedifferentiate? Or what led the cell to dedifferentiate? Certain types of hormones led the cell to dedifferentiate. What are the hormones we are talking about? We are talking about auxin, we are talking about cytokinin. Cytokinin, so obviously we know because cytokinin are most important function key, cell division. Nodule keneke form korile cortex to cell divide kori pelai form korile so which hormone is involved obviously cytokinin right so apart from cytokinin auxin was also involved in nodule formation so question ahe name the two hormones responsible for nodule formation it is auxin and cytokinin apart from that the question the next part who synthesizes auxin and who synthesizes cytokinin. So, a bracket of diya ase auxin is produced by the cortical cells, cytokinin is liberated by bacteria. So, dutai plant dal bacteria duitai duta hormone synthesize koribo because of that hormone the cortex will get dedifferentiated and because of that nodules will get formed. So, nodule formation is dependent upon the secretion of two hormones one by the plant itself the other by the bacteria. Plant ki form koribo auxin bacteria ki form koribo cytokinin baki hini we understand rhizobium can fix nitrogen only in presence of leg hemoglobin etc etc what we can do is we can stop the class here. Next part in the next class. We can take another class, we can wind up the class chapter in this class itself. Ajiu শেষ কৰি দিব পাৰো, next আৰু এটা class ল'ব পাৰো. That will depend on তোমালোকে কিমান absorb কৰিছা. Right? So it's better we take another class. So up till here, today, up till here it's all, right? We'll not discuss anything. As far as the efficiency and all those, we'll discuss in the next Monday's class, right? In the next class that you come for, that is on next Monday, আজি যিখিনি পঢ়িলা, please read and then come to the class. So that if you have any doubts regarding this portion that we have explained, we can discuss it once again, right? Don't just take the note, put it, bring it on next Monday. এনেকা নকৰিবা. ঠিক আছে? আজি আধা কৰিছো. Next classৰ কাৰণে বেছি নাইও, অকমান বাকি আছে. So next part we'll do in the next class. এখিনিলৈকে please study and bring your doubts. And if possible, then see the MCQs also, what has been asked from this part. Right? Then only you will have an idea. Any theory পঢ়ি থাকি লাভ নাই. So that's all for today. Let's not go any further. So up till page number 57, no? So up till page number 57, please study and come for the next class. Okay? Thank you. Okay. 57 লকে পঢ়িবা দেই. Next Monday le. Thank you.